Hello, welcome to Science of the Times. I am Syma Khalid. This week's episode is based on a lecture I gave in Bradford City Hall at the end of May earlier this year. This was the second lecture in an annual series of lectures named after Ibn Sina and hosted by the Muslim Institute. For those of you who are unaware of who he was, Ibn Sina was a polymath. He excelled in mathematics, in philosophy, most notably medicine, and it's for this really that he's most well known. Ibn Sina lived about 1,000 years ago during what is now referred to as the Golden Age of Islam. He lived in what is now modern day Iran. Okay, so now with the permission of the Muslim Institute, I'm going to switch over to the coverage of the lecture itself, in which will cut straight through to where I begin to discuss my own research on computational microbiology, whereby I use computers to try to understand how bacteria protect themselves, how they keep out harmful substances, like, for example, antibiotics, but allow in essential nutrients like vitamins and ions that they need to survive. Of course, watch this or listen to this on any format you like. However, my recommendation is that perhaps you use the YouTube coverage for this particular episode, because there are some visual elements that I referred to during the lecture, which of course you will not be able to see if you have audio only. Okay, so let's switch straight over to the coverage of the lecture. Talk about at some point. Okay, and so now you can see there the stark figures. 1.27 million deaths caused directly by antimicrobial resistant bacteria in 2019, with a further almost 5 million associated with the same issue. Okay, so these numbers are going to just get worse and worse. And the problem is, nobody is trying to discover new antibiotics. There are no pharmaceutical companies really working in this area. So it's called the discovery gap. The last real novel antibiotic was discovered over 20 years ago. There have been one or two since, but it's just been tinkering with already existing ones, no new classes. And the reason for that, if I'm honest, the brutal biggest reason is it's hard to make money. It's almost impossible to make money from making antibiotics. And the reason for that is resistance develops immediately. So if a new antibiotic is discovered, you will not be allowed to sell it and use it, because they'll say keep it for when the pandemic comes. Because the moment you start using it, it stops becoming effective. The resistance begins. And so that's the real driver. And so working on antibiotics, therefore, is left to charities and left to academics. And it really kind of, I guess, motivates a lot of the work I do. Okay, so I just wanted to spend a few minutes sort of explaining to you why bacteria are so hard to kill. You know, you could say we are complicated humans. We have many, many cells. We have brains. We have eyes, nose, mouth, hands. We've made weapons. We've made bombs. You know, why can't we kill a bacterium? What is so hard about it? And so ultimately, this is what we want to do, right? We want to be able to fire antibiotics at a bacterium, and the pathogenic bacterium. I just want to say at this point, we don't want to kill all bacteria. They are good bacteria, the ones in our guts, the ones that live on our skin. We need those, right? We can't process all foods without bacteria. We do need them. And the ones on our skins keep us also safe. It's the bad, harmful ones we want to kill. And the problem is they're protected by a formidable defense system. And it's a little bit like many of you may have seen these concentric castles. So what happens is you breach through the first wall, the defense of the castle, and then you hit with another defense. That could be a moat, so it could be water. You get over the water, and then you're hit by cannon. It's firing cannons back at you. It has multiple defense layers that it's built. And for those of you who are a bit more astute maybe with chemistry will understand what I'm going to say next, is that some of these layers are like oil droplets. Okay, so some of them are like oil and some of them are like water. Now we have a problem. You guys all know that something which mixes with water will not mix with oil and vice versa. Okay, so how do I make a molecule? How do I make a drug? That it will go into oil and then it will get through the oil and then it will get into the water. It will get through the water, then it will get into the oil and then it will get inside. Does that sort of make sense? We have a real problem. They've developed these massive defense systems. And then not only that, they have developed pumps. So if the antibiotic starts to get inside, they have these pumps that start pumping them back out.
And when they sense they're under attack by antibiotics, they start producing more pumps, right? So they are very, very sophisticated. But at the end of the day, there's still no brain, eyes, nose, or mouth. So everything that is happening at the level of individual molecules. And it's this that I'm trying to, not particularly well, I guess, understand. And so I just wanted to focus on some work we've been doing on the very outside defense, trying to understand how this works. I won't go into the details too much, but what I wanted to say is some of you may ask, well, how do you build your computer models? Where does the information come from? That information comes from my colleagues who do go into the lab, who do wear white coats, and they do the experiments, and they come out and they give me information. This could be from microscopes. They say, hey, Syma, this goes here, this moves around here, this works best at this temperature, or I need this much salt, or I need this to get this to work, for example. They might be doing X-rays. So it turns out you can fire X-rays at proteins, just like we can get X-rays if we break our arm or something. We can also get X-rays of proteins and see what they look like. So in my lab, we put all of that together, and we build these huge computer models. And I'll just show you a little movie which is not particularly informative, I guess, and just gives you an idea of the scale of the model. So this is the biggest simulation of a bacterial outer membrane ever done, as far as I know. And this was done in my lab in 2022. And so this was, of course, something we had to do on the supercomputer in Edinburgh. This will have taken about 3 weeks of calculation. So you just let the calculation run for 3 weeks, day and night. Then you get the data out, and then it's analyzed. Okay, so what does it do? What do we learn from it? Whoops. Okay, I just played this movie here. So what we learn is the different colored balls that you can see. These are antibiotics. The balls are flying around, and the blue things are proteins. They're hitting the proteins. You cannot get this level of detail from any experimental method alone. You have to combine experimental methods. And so what we're showing is we can see the antibiotic is getting stuck here, or it's getting stuck here. This is what we need to change to ease its path in. So this is the kind of information we can get from these calculations. Okay, the diagram on the top right, you can see 4 in blue. These are 4 proteins, and the different colored molecules you can see, they are antibiotics. We then go back and we study in detail where is the antibiotic sticking to the protein, and then we can go back and we can talk to our medicinal chemistry friends, and we can say, when you make the antibiotic, change it here or here or here, and then from there we can learn something. And this sort of approach that we use in my lab is very different to the traditional approach. And this is what computational microbiology is. So the traditional approach, you can see a picture there. In blue, you see 1 protein. In yellow, you see 1 antibiotic or potential drug molecule. And this is traditionally what has been done until a few years ago, where we tried to, within my group, we tried to shake the field up a little bit. We sort of said, and I come from a chemistry background, and so I sort of had an understanding of that, and then sort of, when I moved to Oxford the first time round as a contracted researcher, it sort of occurred to me that biology is not the same as chemistry. In chemistry, I have my test tube. I know exactly what I put in there because I put it in. I shake it around and something happens as a function of what I put in that test tube. Biology and nature is not like that. I do not know what is in there. Sometimes I know what's in there, but I don't know what it's doing. And so my philosophy now with my group is to put in as much information as we can, wherever it comes from, even if we think it's not relevant. It may be relevant in a way we don't understand. Right? Nature is not something we can understand so easily. And so our philosophy has been to put in as much detail as we can into the calculations, and something may emerge then in a way that is unexpected. And there was a privilege a couple of years ago that the 30th anniversary edition of the journal Structure then invited a review article on computational microbiology, which I felt like was a bit of a validation that this field is now sort of arriving as something that's being taken, is something that's being taken seriously, and people are thinking maybe this is the way to do things. Okay, so I'm just going to sort of now focus on maybe one of these watery regions. So I said that there were oily regions and then there's watery regions. The watery regions is called the periplasm. It's densely packed with proteins. And so what we're trying to understand in my group is what is interacting with what. And sort of, Adiba said I was going to talk about some of the challenges, and this is a challenge which I'm going to sort of talk to you about now. Here is a movie from one of these simulations. So this was done a few years ago now when I was still at the University of Southampton. It was done by a very talented postdoc who's now moved back to Brazil. And you can see there's a lot going on in this movie. The red stuff is moving, the orange stuff is moving, the bluey bits are moving, everything is moving around. And the water and the salt I've taken out just to make things easy to see, right? And so what we have to do now is, on the one hand, I've said, sort of given myself a pat on the back and said, I'm very clever, I'm putting all of this detail in, no one else is doing that. But I've kind of created a problem for myself. Right? Because now I have to analyze what comes out. So if I build this thing where I put in all my molecules, my antibiotics, my proteins, all the other stuff that I think exists in real life, lots of conditions, different temperatures, different salt conditions, and I try and understand it, this is what the graphs look like. So hopefully you'll agree with me that it's very difficult to tell the difference between the one on the left and the one on the right. Apparently, mathematically, there is a difference. There is some signal in that noise, but to my eyes, they just look like a messy noise, basically. And so what we're doing now is we're collaborating with Arvind Ramanathan in Argonne National Laboratory in the USA. He's an artificial intelligence expert. And what we're trying to do is we're trying to use this kind of thing. So when you guys pick up your mobile phone, and you take a photograph, it recognizes the face, right? All the algorithms now are clever. They know this is a face. There is something in there that recognizes the signal. These are two eyes, this is a nose, this is a mouth. Can I use the same technology to find the signal in all of this noise in my data? And it turns out we probably can. So it's a very fruitful collaboration that we have with Arvind. And just sort of to take that on one step further, this is some new stuff that I'm really excited about. Actually, the last is quite timely because the very newest data just came out 2 days ago from this. What we've set up with Arvind is we do the computer simulations in my laboratory in Oxford. We then email the results directly to the robot in Argonne. The robot makes the antibiotic, tests it on a plate with the bacteria, then does an image analysis, how many are dead, how many are alive, feeds that into our artificial intelligence algorithm, improves the design of the antibiotic, back to my laboratory for more simulation to say we think this is how it works, then back to the robot. So this is just something we've been set up in the last few days. And I guess ultimately the hope is that you can take me out of the equation. At some point, the artificial intelligence will learn enough that you no longer need us to do the simulations. It will have enough to carry on and improve without us. But for now, it needs our mechanistic insights. And then to kind of finish off, just to answer one of the queries you put at me about what the future might look like. Well, I think some of the questions we perhaps should ask going forward is about sort of, even if I work out an antibiotic, why is it that one antibiotic might work for me but not for you? Why is this? And oftentimes, why is it that some bacteria will be effective, some antibiotics will be effective against this bacterium but not this one? Why is that? You know, and sometimes when you go to the doctor and you have a bacterial infection, it takes them 2 or 3 goes to get the right antibiotic for you. Why is that? And I think these are the kind of questions that we should be thinking about now moving forward. And it turns out that this is within the realm of something called personalized medicine. So we're almost at the stage where it's beginning to become possible to deliver personal medicine tailored for individuals. And this really comes from enormous steps in genetics and our ability to sequence the human genome. So to find the secrets which are within our genomes. So for example, my brother is in the audience, we have the same parents, but what makes him different to me? right? So this secret is in our DNA. And for example, I have friends and colleagues at Oxford. Nanopore Technologies, a wonderful world-leading company that's come out of Oxford, and they are now managing to do this. You can do it on something as small as a USB stick now. They can take that and they can sequence genomes using this technology. And I think that's the future. The future is going to be not just a sort of a widespread generic medicine, but a tailored, personalized medicine. And so with that, I kind of want to leave you with an outlook that from my side, my work, and sort of I can see in the next few years, what we're trying to do is we are trying to use our simulations as computational assays. So the doctors, the white coat wearing scientists, they do assays. I want to use my computer to do assays, but to do that, I have to put in as much detail as I can. I firmly think that. I think it's not sufficient just to say, here's my drug, here's my target, this is what I'm going to simulate. I have to put in everything else as well. And that's not so easy to do because sometimes we don't know what's in there. But we can keep moving and going forward and getting information from as many collaborators and people who are willing to provide that as possible. So that's sort of what I wanted to say about the science. But what's also incredibly important to me is, and I think this also chimes with the ideals of the Muslim Institute, is to spread the word of science, to spread the word of bringing science to wider communities. And if you might indulge me for a second, I think oftentimes, our communities have been less willing to do that because oftentimes we want our children to be doctors. But how many of us want our children to be scientists, the ones who make the medicines and discover them so that the doctors can apply them? And I think hopefully the pandemic, if anything good has come out of it, some of the things can be that this learning is also incredibly important. And so to spread some of this knowledge, one of the things I've been doing recently with my colleague Tim Coulson at Oxford is we've set up a podcast. It's called Science of the Times. And you can find us on YouTube, Spotify, Apple podcasts. And what we try to do is bring science stories to the wider public in a way that's not patronizing, but still accessible. And so if anyone wants to know any more about that, give me a shout. I'd be more than happy to chat to you. And I bought some flyers if anyone's interested. And indeed, if you have ideas of science stories or there's things about science that you want us to cover, that you want to know more about, then please email in. We're looking for engagement, actually. And with that, I want to acknowledge the people. So it's my privilege to stand here, sit here and talk to you about the science. But of course, I don't do the science. The science is done by my team. And so I acknowledge those members of my team who are with me right now at Oxford. But of course, the work I've presented has come over 20 years of research, and I can't possibly name them all. So this has included undergraduate students, PhD students, postdoctoral researchers, and I'm grateful to every single one of them for their diligence and hard work and for putting up with me as a crazy at times boss. Also collaborators, none of us is an island, no work is done in isolation. I have wonderful collaborators all around the world. Some of them are computational, some of them are experimental. And what I love about my collaborators is when we don't agree, they never say the calculations must be wrong, right? I never say the experiments must be wrong. We always think maybe we've measured the wrong things. So let's meet in the middle. And that open-mindedness has been wonderful. And every single one of those collaborators is the same. And finally, I want to also acknowledge my funders. Science is not possible without money, I'm afraid. And so the ones named at the bottom have supported my research for many years. And Biosim is this organization that provides computing time on the national resource in Edinburgh. And this is my final slide, but before finishing, I also wanted a special acknowledgement really for my support team, and if you like, my dream team. Here they are, and from the bottom of my heart, thank you. And thank you. Thank you.
Hey, welcome back. I hope you enjoyed the lecture. I must say it was an exciting and at the same time nerve-wracking thing to do. The surroundings were beautiful. Bradford City Hall really is a wonderfully elaborate and beautiful building. The Lord Mayor of Bradford was in attendance. It was a great audience. Unfortunately, what we don't get in the coverage is the question and answer session. There were some really probing questions about the use of artificial intelligence in the discovery of therapeutics, the threat of antimicrobial resistance, and also some more personal questions in terms of any impediments or barriers I faced in my own career due to, various ticking various minority boxes, I guess. And if anyone is interested in any of my responses to those, then please do get in touch. I'd be happy to share that with you. I must say, what was most nerve-wracking for me was the fact that my parents and my brother were also in attendance. And when it got to the question and answer session, I was petrified that my dad would hold up a standard plug and ask me if I know how to change the fuse in that. But luckily, he desisted, and that didn't happen. It was in the end, it was a wonderful event, and I'm very grateful for the Muslim Institute, and of course for you for listening and for your attention. As always, if you liked what you've seen and heard here, then please do subscribe, tell your friends, send us some comments, and any ideas you have for future episodes. Okay, guys, until the next time when Tim will be back, take care.